The destruction of humanity began, as many things do, with the best of intentions. The artificially created retrovirus known as ALZ-112 was initially designed to target and repair damaged neurons in humans affected by Alzheimer's, an admirable goal to be certain. But as the world has seen, the ultimate results of this virus were not as the creator had hoped. Instead, this cure led to the complete collapse of human civilization, with a death toll in the billions. But this virus had another effect. Whereas infected humans died outright or eventually lost their higher cognitive function, apes infected by this virus exhibited increased intelligence, so much so that they moved from merely sentient to sapient. Now, allow me to describe in detail the biological mechanisms that forever changed the world as you knew it, from the virus's interactions with its very first hosts to the mutations that would destroy what was left of humanity, even as it uplifted the apes. But I warn you, though it may sound fantastic, it is not as far-fetched as it may seem. But before I describe the effects of this virus, I must describe the insidious disease it was meant to eradicate: Alzheimer's. Perhaps the most broad characterization of Alzheimer's disease is progressive cognitive decline due to the degeneration and death of brain cells. This degeneration is primarily associated with the accumulation of amyloid beta plaques outside the neurons and tau protein tangles within the neurons. Amyloid beta plaques disrupt synaptic communication between the neurons, while tau tangles inhibit nutrient transport within neurons, leading to cellular dysfunction and death. With the noble intention of countering these pathological processes, in 2011, a San Francisco-based biotechnology company known as GenSys Laboratories began to develop an artificial retrovirus known simply as ALZ-112. More specifically, this genetically engineered vector was designed to deliver therapeutic genes directly into neurons, facilitating the breakdown of the amyloid beta plaques and preventing the formation of tau tangles, in theory thereby restoring and maintaining normal neuronal function. At least, that was the idea. And for a time, it seemed to work. In animal models, particularly primates, the results of ALZ-112 were promising. But of course, simian testing could only tell us so much, and one scientist began human testing, beginning with a single subject afflicted by Alzheimer's. In fact, much as was observed in primate testing, this human subject showed a promising improvement in cognition, including memory retention. Sadly, however, these effects were short-lived. The human immune system, recognizing the viral vector as a foreign entity, responded by producing antibodies against ALZ-112, neutralizing the therapeutic effects of the virus and leading to a complete cessation of its neuronal reparative action. And within a short time, the subject had entirely reverted. It was this failure to achieve lasting results with ALZ-112 that prompted the development of a second strain, this time engineered to evade the human immune response. In fact, this new version, called ALZ-113, was not only designed to resist immune detection, but also to increase the efficiency and permanence of its genetic alterations. But as we know, the modifications that were intended to enhance the virus's resilience and its capacity to integrate therapeutic genes into the human genome actually increased its potency and entirely altered its impact in both humans and apes. You see, due to its nature as a retrovirus, ALZ-113 operates by integrating its genetic material into the DNA of the host cells, which in turn is facilitated by the viral enzyme reverse transcriptase. This enzyme essentially converts the viral RNA into DNA and subsequently integrates it into the host genome. This process is important not only for the virus's replication, but also for the long-term expression of its genetic payload, which includes the therapeutic genes initially intended to counteract Alzheimer's pathology. In humans, the integration of ALZ-113's material led to highly unpredictable genomic interactions. It appeared that the integration of delivered genes sometimes occurred at sites critical for normal cellular regulation, causing a disruption that led to the misregulation of native genes and even the activation of oncogenes, contributing to widespread cellular dysfunction and severe immune reactions. Worse, the virus's surface proteins had been engineered to minimize recognition by human immune cells, allowing it to spread far more efficiently and to infect a larger number of cells before being detected. In short, this highly contagious virus caused catastrophic mortality rates, with the primary symptom being hemorrhagic fever. And when containment of the virus failed, hospitals and emergency rooms all over the planet soon became overwhelmed with patients showing signs of ALZ-113, now also referred to as the Simian flu. Transmission of the Simian flu was quickly determined to occur primarily through respiratory droplets and close contact. Once symptomatic, individuals could spread the virus through coughing, sneezing, or even simply talking. Within 48 hours, many infected individuals experienced severe headaches, sore throat, fever, and joint pain. At the 98-hour mark, symptoms included nausea, vomiting, raised rash, chest pain, and severe cough. The final stage of symptoms occurred at 7 days, with most victims experiencing stomach pain, severe weight loss, and bleeding from all facial orifices. Tragically, only 1 in 10 of those infected survived, and within a mere 8 months, as many as 150 million humans were dead. Worldwide anarchy followed rapidly. And within 5 years, the human death toll had risen to a staggering 50% of the Earth's population. Intergovernmental organizations as well as the CDC attempted to develop a cure, but were entirely unsuccessful. A complete breakdown of society and government would follow, and within a decade, the worldwide reign of humanity had come to an unceremonious end. In apes, however, the same genetic alterations that proved devastating to humanity actually enhanced cognitive and physiological functions, granting them heightened intelligence and eventually sapience. And indeed, this process is both fascinating and unprecedented. Once the virus has made its way into a simian host, spread in the same manner as in humans, the virus begins to multiply and disseminate throughout the body. Then, due to the engineered properties of ALZ-113, the virus crosses the blood-brain barrier, which normally protects the brain from pathogens. How this is accomplished is unknown to me, but it seems likely that the virus hitchhikes inside the migrating immune cells, a process known as Trojan horse entry. Once inside the brain, ALZ-113 binds to specific receptors present on neuronal cells, and crucially, and as mentioned previously, begins to integrate its genetic material into the host's DNA, in turn drastically altering neuronal function. And this is when the primary action of the simian flu takes place. As the viral DNA is integrated into the neurons, it appears to prompt the overexpression of genes involved in the birth and differentiation of new neurons, as well as synaptic formation and plasticity, the latter of which regulates the strength and efficacy of synaptic connections. As a result, infected apes begin to exhibit greatly improved memory retention, problem-solving skills, and spatial awareness. Within a few weeks, apes routinely display behaviors indicative of higher cognitive processes, such as strategy formation and even abstract thought. But the neuronal changes aren't limited to memory and cognition. Remarkably, as these changes are taking place, several regions of the brain associated with language and motor control are also greatly enhanced. First, when the viral DNA integrates itself into the brain, both the motor cortex and the areas analogous to the human's Broca's area, which coordinates the muscles involved in speech production, show increased functionality and connectivity. Now, even before infection, many great apes are capable of understanding gestures, even interpreting them to form limited abstractions. The idea that the neural circuitry for higher communication might already exist in apes, albeit in a less refined form than in humans, had long been thought possible. And indeed, it has been observed that chimpanzees, for example, intentionally produce manual gestures as well as vocal signals in communication with humans. In short, prior to the outbreak, it had been suggested that in apes, the precursors to human language are present at both the behavioral and neuroanatomical levels. by enhancing the expression of certain genes specifically involved with the ventrolateral prefrontal cortex, which comprises the Broca's area in humans. ALZ113 effectively boosts apes' capacity for auditory processing, as well as by repurposing the neural substrates originally adapted for gesture recognition. I should note that this region is also associated with certain motor functions, resulting in greatly improved orofacial control as well as the management of respiratory rhythms associated with speech in humans. But of course, in apes, the cognitive ability to communicate was not the only obstacle in achieving speech. As we've established, the capability of communication was evident in apes even before the outbreak. Vocal speech, however, was not possible for a few reasons. It was long thought that apes and monkeys simply lacked the appropriate vocal anatomy altogether, despite being able to produce a limited and inarticulate range of sounds. But in fact, many species did possess the ability to enunciate to a certain degree. The real issue was that most primates simply lacked the neural control over their vocal tract muscles to properly configure them for speech. In other words, given an alternate and more effective neural configuration, such as what we see in humans, many primates could talk. By targeting the nerves controlling the larynx, as well as the motor cortex previously described, this is precisely the neural configuration that ALZ113 accomplished. But the virus didn't stop there. Many primates' vocal anatomy includes membranes, also called vocal lips, that are effective in producing loud, resonant calls, a crucial adaptation for long-distance communication across distance. However, these membranes can make it difficult to control vocal pitch and volume with the precision required for speech. Intriguingly, the virus appears to downregulate genes responsible for the development and maintenance of these vocal membranes, altering their expression in a way that leads to their eventual regression. Regression is also observed in the cavities that branch off the main vocal tract. These air sacs serve several functions, including enhancing the volume of calls without the need for more rapid breathing, simultaneously preventing hyperventilation during extended periods of calling. Once again, however, these air sacs interfere with the subtle modulations in air pressure and flow that human speech requires, and in humans they are greatly reduced. Finally, the muscles attached to the vocal cords, such as the thyroarynoid, cricothyroid, and arytenoids, undergo significant changes as a result of ALZ113, increasing in both elasticity and responsiveness, and allowing the vocal cords to adjust more rapidly and with greater precision. The overall result of these changes is greatly improved pitch modulation and phonetic variability, each of which, combined with the neural changes mentioned previously, bestows upon infected apes the ability to produce speech in a manner strikingly similar to humans. One final point of note is that the simian flu appeared to alter even the eye color in infected apes, changing the iris to a distinctive green. Though the precise mechanisms of this change aren't yet fully understood, it appears that the virus influences the genes that regulate melanin production in the iris by inadvertently affecting those that regulate pigmentation, such as OCA2, and leading to a new phenotypic trait. Interestingly, the changes wrought by the simian flu do not only affect those apes that are directly infected. Indeed, because the virus integrates its genetic material into the germline cells of the initial hosts, the organism's modified genes are passed down to subsequent generations, embedding the new traits, such as the neural and anatomical changes described, within entire populations. And so it was that a new planetary ruler emerged, as other primates replaced humans as Earth's dominant species. Perhaps tragically, all of this occurred as the result of humans' own tampering in realms no one at the time could have fully understood. But the simian flu didn't just uplift certain primates and prove almost universally fatal to the human population. Nearly 2 decades after the outbreak, humanity still clung to life. But ALZ113 was about to change. While the simian flu proved fatal to nearly the entire population of Earth, it quickly became apparent that some humans, though infected, were immune to the virus's most devastating effects. The reason for this immunity is not certain and would require a number of specimens for study, to which I have little access. Still, we can speculate. It seems likely that the genetic variations in certain human individuals could include differences in the receptors used by the virus to enter human cells, or variations in the immune system's ability to recognize and resist the virus in the first place. Whatever the case, the result is that humanity did survive, albeit in small, isolated pockets. Approximately 15 years after the initial outbreak, ALZ-113 mutated. Unfortunately, scientific studies at the time were severely lacking, and the exact mechanism of this mutation is unknown. It was likely inevitable, however, as the method of reverse transcription employed by many retroviruses is inherently prone to errors. However it occurred, this mutation led to a strain that was less deadly to humans by far than the original, and did not appear to negatively affect non-human primates. But while it was no longer fatal, for humans, the new effects of the virus could be considered even worse. Though some limited immunity is still present in certain rare individuals, in general, infected humans affected by this new mutated form of ALZ-113 now exhibit a drastic reduction in cognitive abilities, as well as the complete loss of the ability to speak, much less communicate effectively. Unlike the original virus, which had a widespread effect on neural tissues, the mutated strain exhibits a form of selective neurotropism, preferentially targeting specific regions in the human brain, such as the Broca's area, Wernicke's area, and the prefrontal cortex, regions responsible for speech production, comprehension, and higher reasoning, respectively. It is in these regions that we observe the most drastic changes from the original strains. For example, in a staggering reversal from the intended therapeutic design, instead of enhancing neural connections and promoting neurogenesis as ALZ-112 did, the mutated virus actually disrupts neural signaling, leading to the poor regulation of proteins involved in synaptic function. This results in protein misfolding, dysfunctional neuron signaling, and eventually cell death. And because regions such as the Broca's area directly influence the neuromuscular control of the larynx, throat, and other vocal apparatus, even the ability to physically form words is deeply negatively impacted. In short, humans who once thought themselves safe from the most devastating effects of simian flu soon found their very intelligence slipping away for reasons they surely did not understand. Worse, just as the improvements in cognition in apes were found to be hereditary, so too were the deleterious effects in humans. Indeed, generations of humans were now born with inherent cognitive and speech impairments, further cementing the apes' planetary rule. At this point, you may be wondering, as I did, why is it that the effects of the virus are so drastically different in humans and apes, both of which are classified as primates? Indeed, this was precisely the focal point of my research for some time. Let's begin with the first aspect of this overarching question. As we know, ALZ-113 greatly increased ape intelligence while simultaneously proving fatal to 90% of humanity. This presents two sub-questions, so to speak. First, why did the virus not also increase human intelligence? The answer is simple, though admittedly speculative. It seems likely that those who were immune to the virus's deadly effects were also immune to its improvements, so to speak. The second question is the inverse. Why did ALZ-113's initial strain not kill its simian hosts? The answer to this question is less simple, but it likely stems from fundamental differences in how ape and human bodies respond to viral infections, particularly the slight variations in the structures of cellular receptors. In humans, ALZ-113 triggers a severe inflammatory response, leading to the aforementioned symptoms of hemorrhagic fever, such as vascular leakage and multi-organ failure. In contrast, apes exhibit a more regulated immune response that effectively contains the virus without causing excessive tissue damage. Furthermore, in apes, we see a lower cytokine response, preventing the cytokine storm associated with the same infection in humans, thereby reducing the likelihood of hemorrhagic symptoms. This helps explain why the initial strain of ALZ-113 affected the two groups so differently. But when it comes to the mutated strain, now referred to as ALZ-113M, an even more fascinating difference between humans and apes began to emerge. As we've already established, following the outbreak of the simian flu, 1 of every 10 infected humans proved to be immune. Of course, even those who exhibited no symptoms were still presumed to be carriers of the virus. No such statistics were measured for apes, though symptomatic rates are quite high as well, and immunity appears non-existent. By the time the virus mutated, the only humans in existence had not only avoided the fatal effects of ALZ-113, but also its cognitive effects. Apes, on the other hand, had been universally affected, their brains essentially rewired for greater intelligence. It was precisely these conditions that laid the groundwork for the results of the mutated strain. Because ALZ-113 not only boosted apes' cognitive abilities but also enhanced the plasticity of their neural networks, the ape brain became both more adaptable and more resilient to subsequent changes, including those induced by a mutated virus. Furthermore, over time, ape physiology had adapted to the continuous exposure to the virus, leading to the upregulation of protective genes, as well as neural pathways that compensated for the negative effects brought on by the mutated strain. Not only this, but it also appears that apes developed a form of immune tolerization, wherein their immune system learned to manage the virus without launching an overly aggressive response that could damage the brain, even as the virus mutated. Extant humanity, on the other hand, had no such biological priming. Their own brains, especially in the aforementioned regions of language processing and communication, had not developed the enhanced regenerative abilities or the plasticity observed in apes. And without previous exposure having led to adaptive changes in the brain, humans were more susceptible to any alteration in the virus's behavior. In short, when ALZ-113 mutated, these humans simply lacked the neuroimmunological adaptations necessary to mitigate the effects of the virus on both cognitive functions and speech, causing the near-complete degradation of the once great human species. It is perhaps little surprise that humanity would be the source of its own downfall, though the manner in which it occurred could never have been predicted. But no matter the cause or the means, the current state of humanity, and indeed their overlords, is unlikely to change in the foreseeable future. Ape society will continue as it has now for hundreds of years, spreading like wildfire throughout the world. At the same time, the dying embers of humanity, though they may occasionally crackle with a spark of intelligence, will soon grow entirely cold.